Hello everyone, this lecture video is all about gluconeogenesis. And here, gluco, it means glucose, neo, it means new, and genesis means creation or beginning. So we can define this as creation of new glucose. And we can further define this as a metabolic pathway by which glucose is synthesized from non-carbohydrate materials. Take note of the word non-carbohydrate materials because this process or pathway creates glucose from non-carbs, meaning to say it creates glucose from fats, amino acids, proteins, and reversible lactate, which is one of the fates of pyruvate. So if you can remember, from the previous lecture, I have mentioned that glycolysis is a irreversible process or a linear pathway because it cannot regenerate the glucose itself. Every day you intake carbs, and this carbs is being converted readily into glucose, and our body only needs 100 to 200 milligrams per deciliter of glucose. So if you intake too much carbs, there will be an overproduction of glucose, so there will be an excess glucose, and the excess glucose will be stored in liver as glycogen. Here is the glycogen, and that will be stored here in the liver, and this glycogen here will be utilized or will be used when blood glucose drops. The drop in blood glucose level usually happens when you are in fasting mode, let's say intermittent fasting, in which you will eat for 8 hours and you will only drink for the remaining 16 hours. And it also happens when you are in a sugar-free diet, let's say you are in keto diet, which means you're only eating protein, then the stored glycogen here will be depleted in the long run because you are being utilized that and you are not intaking any carbs. So when this happens, gluconeogenesis will occur because some of the tissues such as nerve tissues or neural tissues and exercising muscles or red blood cells only utilizes glucose as energy source. So if you have not intaken any carbs, gluconeogenesis will occur to produce glucose from non-carbohydrate molecules such as fats, amino acids, and lactate, which is one of the fates of pyruvate. This one will be converted back into glucose in the process of gluconeogenesis. But in glycolysis itself, it cannot be reconverted into glucose. So, the main function of gluconeogenesis is to maintain the blood glucose level because, again, the nerve tissues and RBCs, for example, only utilizes energy from glucose. And gluconeogenesis takes place in liver, kidney, and the small intestine. So, this is the pathway for glycolysis and for gluconeogenesis. This is for glycolysis here. This is a linear pathway and irreversible one because this pyruvate here, okay, cannot be converted back again in glucose in glycolysis. It cannot regenerate the glucose with the same enzymes here. So gluconeogenesis has to occur for us to regenerate the depleted glucose or depleted glycogen if you weren't able to intake any carbs. But if you are continuously intaking carbohydrate, there's no need for gluconeogenesis to occur. And then let us start. This one is an irreversible process because as you can see here, the enzymes here that is being used in the production and in the breakdown is different. When you say they are reversible, the enzymes that are being used must be the same both in the synthesis and breakdown. Let us start here with lactate, which is one of the fates of pyruvate. In gluconeogenesis, you can convert lactate into pyruvate with the help of the enzyme Lactate Dehydrogenase. It will remove hydrogen here and that will be converted into pyruvate. Then this pyruvate here, with the help of the enzyme pyruvate carboxylase, will convert the pyruvate into oxaloacetate with the help of the ATP and carbon dioxide gas. So such as this one. This is the chemical reaction for that. The pyruvate here, with the aid of the carbon dioxide and ATP plus a water molecule, and with the help of the enzyme pyruvate carboxylase, which is being released by biotin, which is a microorganism that is settled in our small intestine. Pyruvate carboxylase, meaning to say it will add carboxylate ion here in pyruvate, unlike the decarboxylase, which will remove the carboxylate ion. The carbon dioxide gas here will become a carboxylate ion here that will be attached here. The CH3 will become CH2 since you used a bond here. So you have now your oxaloacetate, and the ATP because this requires an energy to be performed. So the ATP will become ADP, and there will be an inorganic phosphate that will be released from the ATP. So that is the process for the pyruvate to oxaloacetate in gluconeogenesis. And then, once that is being converted into oxaloacetate, it will be converted into phosphoryl pyruvate with the help of the enzyme phosphoryl pyruvate carboxygenase. This is that. Oxaloacetate to phosphoryl pyruvate, addition of JTP with the help of this enzyme. This will be removed and will become a carbon dioxide gas, and the phosphate here will attach here on the second carbon, and there will be a single bond because the phosphate has been attached, and the bond here, instead of the two here, the one bond here will be used to connect the phosphate here. And then the JTP will become JDP, the carboxylate ion will become carbon dioxide gas. So that is, now you have the phosphoryl pyruvate. Once it has been converted into phosphoryl pyruvate, that will be converted in 3-phosphoglycerate. And the 3-phosphoglycerate will be converted into 1,3-biphosphoglycerate. Notice that the enzyme that has been used here are the same, but here on this case, no ATP is required because here in glycolysis, this is a breakdown of phosphate. We have 2 phosphate here, right? So with this help of this enzyme, a phosphate has been removed. Okay, you only now have the phosphate in 3-phosphoglycerate. But here in gluconeogenesis, when you convert the 3-phosphoglycerate into 1,3-biphosphoglycerate, you will need an ATP because you will add a phosphate on the first carbon. So you now have the 1,3-biphosphoglycerate here. Then this 1,3-biphosphoglycerate here will be converted into fructose 1,6-biphosphate. And the fructose 1,6-biphosphate will be converted to fructose 6-phosphate. So again, notice here that the phosphoproctokinase here in glycolysis that initiate the conversion of fructose 6-phosphate to fructose 1,6-biphosphate requires an ATP because there is only one phosphate that is located on the 6th carbon. But here, there are already 2 phosphate. The 1 phosphate from ATP, the ATP will be converted into ADP, right? So phosphate will be added on the first carbon. That is why you have now fructose 1,6-biphosphate. Here in gluconeogenesis, there is no ATP required. See the difference? Because here, there will be a breakdown of phosphate or removal of phosphate, and then energy, of course, will be produced. So this enzyme fructose biphosphatase, this will remove the phosphate here in first carbon. So you now have the fructose 6-phosphate. This fructose 6-phosphate will be converted into glucose 6-phosphate. Then the glucose 6-phosphate will be converted into glucose with the help of the enzyme glucose 6-phosphatase, which will remove the phosphate in the 6th carbon. And that is why you now have, this will be removed. So that is why you now have the glucose here. So no ATP required as well, because here you will be removing a phosphate, so that is a production of energy. But here in glucose to glucose 6-phosphate, an ATP is required because you will place a phosphate on the 6th carbon of the glucose here on the glycolysis. But here, no ATP required in gluconeogenesis. That is the pathway for the conversion of lactate to glucose. Now let us look on other non-carbohydrate materials. Let us have here the propionate. This one. This propionate here is a lipid molecule, one of the constituents of fats. So this propionate will be converted into succinyl-CoA, and then the succinyl-CoA will be converted into oxaloacetate. Again, this is a Krebs cycle intermediate. So it will undergo the same process or the same pathway as the conversion of the lactate to glucose once it became an oxaloacetate. So it will have the same pattern for the propionate. Now for glycerol, as one of the components of fatty acid, the glycerol will be converted into glycerol 3-phosphate, and the glycerol 3-phosphate will be converted into dihydroxyacetone phosphate. Dihydroxyacetone phosphate is one of the components of glycolysis. So this dihydroxyacetone phosphate here will be converted to fructose 1,6-biphosphate. Once it has been converted into fructose 1,6-biphosphate, it will follow this pathway that has been followed by lactate and propionate to be converted into glucose. So that is the pathway for gluconeogenesis for non-carbohydrate materials and for the lactate, which is irreversible in glycolysis but is reversible. The only phosphates and pyruvates that is reversible in gluconeogenesis. Then here are the stages in which I summarize the two of the most important stages in gluconeogenesis. So, this is the last step of glycolysis, right? The conversion of phosphoenolpyruvate with the help of the enzyme pyruvate kinase into pyruvate. And this is the first two steps of gluconeogenesis, the conversion of pyruvate once the non-carbohydrate or the lactate had been converted into pyruvate that will be converted into oxaloacetate with the help of the enzyme pyruvate carboxylase, which will add a carboxylate ion in pyruvate. And then this oxaloacetate will be converted into phosphoenolpyruvate with the help of this enzyme, phosphoenolpyruvate carboxykinase. So these are the important steps showing the contrast of glycolysis here and gluconeogenesis. The end here will be the starting point for gluconeogenesis. And let us talk about the conversion of amino acids into glucose. Then we have here the glucogenic amino acids, which are represented by color red, which forms pyruvate, alpha-ketoglutarate, succinyl-CoA, fumarate, or oxaloacetate upon transamination. Then we also have here the green ones, which are the ketogenic amino acids, which form acetoacetate or acetyl-CoA. Then we also have here the violet ones, which are amino acids that contain both the properties. So these amino acids here needs to undergo transamination. When we say transamination, that is the removal of amino acid group so that they can be converted into ketone bodies such as the pyruvate, oxaloacetate, and fumarate, and succinyl-CoA. So the enzyme that catalyzes the transamination or the removal of amino group in amino acids is the transaminase. This is the enzyme. So, for them to be converted in these forms, until succinyl-CoA, until fumarate, into acetyl-CoA, into pyruvate. So, these amino acids here will be converted into pyruvate. And when it has been converted into pyruvate, that will be converted into oxaloacetate. Once converted into oxaloacetate, that will be converted into phosphoenolpyruvate. And it will follow the same path here once they had been converted into phosphoenolpyruvate, the same pathway. And then, so these amino acids will be converted into acetyl-CoA. And once it has been converted into acetyl-CoA, it will be converted into citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, oxaloacetate. And once it became an oxaloacetate, it will be converted into phosphoenolpyruvate. So once in phosphoenolpyruvate, it will again follow the same pathway here from phosphoenolpyruvate up to glucose for those amino acids. And these amino acids here, it will be converted first into glutamate, which will be converted into alpha-ketoglutarate, which will be converted into succinyl-CoA, which will be converted into succinate, fumarate, malate, oxaloacetate, and which will be converted into phosphoenolpyruvate. And once it reaches the phosphoenolpyruvate, it will follow the same pathway such as others, phosphoenolpyruvate up to glucose. And then these amino acids here, threonine up to valine, will be converted into succinyl-CoA, and this succinyl-CoA will be converted into succinate, fumarate, malate, oxaloacetate, and phosphoenolpyruvate. So once these amino acids have been converted into phosphoenolpyruvate, they will follow the same pathway as other non-carbohydrate materials, from phosphoenolpyruvate up to glucose using the enzymes here. And then these amino acids here, tyrosine, phenylalanine, and aspartate will be converted into fumarate, will be converted into malate, oxaloacetate, and into phosphoenolpyruvate. And once again, if these amino acids had been converted into phosphoenolpyruvate, they will follow again the same pathway such as other non-carbohydrate materials, from phosphoenolpyruvate until glucose, in which their pathway will be catalyzed by these enzymes as well. And then this enzyme, aspartate and asparagine, will be converted into oxaloacetate first, and once had been converted into oxaloacetate, that will be converted into phosphoenolpyruvate. And once again, when it has been converted into phosphoenolpyruvate, it will follow the same pathway as other non-carbohydrate material that is catalyzed by this enzyme. That is for gluconeogenesis in conversion of amino acids into glucose, which is amino acid is a non-carbohydrate. So that is all for gluconeogenesis, or the production of glucose from non-carbohydrate materials.